Okay, here we go. Video number 6, last one in the bunch. Now that we're DNA experts and know all about transcription, translation, replication, it's time to play with DNA and look at some of the weird tricks that can be done with that. So let's back up for a second because it's been a while and how we figured out, quote unquote, what DNA is and how it works. Remember Chargaff and analysis, and Rosalind Franklin and Watson and Crick figuring the makeup of a DNA molecule. A's equal T's, C's equal G's. Rosalind Franklin and being able to extract DNA. And I've included a link here that we're going to skip over for this video, but shows you how easy it is to extract DNA from any organism. Because remember, all organisms have DNA. And very, very simple, very easy to do. And really, that we know that what we've seen so far, that DNA is amazingly simple in all organisms. Now, because it's in all organisms, that leaves us with the ability, and because it's so simple in its construction and how it works, and this uniformity across all life, that we can do some tricks with DNA. And some of them are, although all of them are rather simple, we'll be doing a few of them in class, some of them have amazing repercussions in what they can do. We can, for example, cut up DNA using special enzymes. We'll be looking at those later in class. Enzymes that actually will recognize specific sequences no matter where the DNA comes from, whether from a human, a tree, a bacterium, a fish, you name it, and it'll cut up that DNA in a similar manner. And because we can do that, because organisms have the same DNA essentially, not the same sequences and the same genes, but made of the same basis, we can mix DNA from different organisms. We'll be looking at how to do that. And we're really unlimited actually in what we can do, because we have the ability to cut and mix and match DNA. Some of the things we'll be looking at that we can do with them, solve murder cases, if you will, or at least from a biology standpoint, compare DNA so others can solve murder cases. Kind of fun. Parental disputes, we'll be looking at that one. That's Jerry Springer-ish. We can create what's called transgenic organisms and mix DNA of various organisms. Take a gene from a lightning bug, for example, and put it into a plant. Kind of crazy. We can mass produce proteins in medicines because we can insert genes into bacteria, allow them to reproduce prolifically and make proteins, and also medicines for us, kind of tricking them into being slaves. Very interesting. And in essence, we can really alter existing organisms. We can create, we'll be looking at this lastly in class through ethics and biotechnology, create genetically modified organisms, or GMOs, which seems good on the surface, but maybe not always good. So just as a quick example to get us warmed up and get us thinking before we look at these simple details, what would something like this tell you? What is this thing? What are we looking at here? I see some lines, I see some smears and some bands, and what is this madness? And we're going to look at looking for patterns and similarities. Notice that this column here matches this column here. That's going to be able to tell us something. We'll be able to make some assumptions from that. But in essence, so what? What do we do with this stuff, and how do we do it? So let's start with our basics. Let's say we've pulled out DNA, extracted DNA, according to that video link, from an organism, maybe from a human, or we'll start simple. Let's say from a plant, such as from a strawberry. We have that DNA. Now, what do we actually do with that? And our starting point, in order to do any trick, is first we have to cut the DNA. Now, we're not going to use scissors here. Remember, DNA is microscopic, very, very small, packed, although very lengthy molecule, simple in its makeup, and packed into a nucleus. So we can't see it so that we can cut it specifically. We're going to employ specific enzymes, what are called restriction enzymes. Remember, enzymes make a chemical reaction work. And in essence, all they're going to do is cut DNA in certain spots. They're going to recognize specific sequences and cut it at that site. So we'll look at what that is in a second in a video here, but in essence, here's my double-stranded short little sequence here. Notice my A's with T's, C's with G's. But an enzyme just like DNA polymerase and helicase, or in a similar manner, is going to recognize a certain sequence in here, and it's going to cut it, not in a straight up and down manner. It's going to cut it in a staggered manner. So notice this enzyme, whatever it is, recognized the sequence AATT, and it cuts the DNA so that we have a staggered, overhanging little single-stranded fragments called sticky ends. This TTAA should match up with AATT, but that enzyme broke those hydrogen bonds and the phosphate sugar backbone. So now instead of one piece of DNA, we've got two smaller pieces, both of them with sticky ends, fragments, if you will. Pretty simple. So now let's really quickly look at some aspects of another video here, this recombinant DNA technology, which is really just saying how we can cut this. So for example, this circular piece of DNA is a bacterial plasmid. Here's a gene that we want to insert in that. But if we cut that DNA and cut this DNA, we're actually able to mix the two DNA molecules together in a very, very simple manner in doing so. So let's zoom in and look at this. Remember, this is a DNA molecule here, double helix. If we untwist this to look at it, A's and G's and C's and T's. What we're going to attempt to do is cut this. So here comes our restriction enzyme, has a weird name, PST1, which is named after the organism was discovered. And just like a pair of scissors, it's going to recognize and cut that. But when it does cut it, it cuts it in a staggered manner. Remember again, so that we have actual sticky ends on there. And those two sticky ends can actually now form bonds with another piece of DNA, as long as that other DNA has on its sticky ends two matching opposite pieces. Notice that our overlapping ends, an enzyme will come in and seal that shut. So we now have a piece of DNA with a new gene inserted into it. This gene has now become part of the DNA of that organism. Kind of crazy when you think about it. So now that we've cut the DNA, again, we can do whatever the heck we want with that. In addition to actually inserting and mixing genes together, we also can separate the DNA to compare that. And the process to do that is something we're going to be doing in class together called gel electrophoresis. Now that's a fancy name, but essentially all we do is take these samples of DNA that have been cut into fragments and we insert them into a gel. And we give it a little bit of electricity, as we'll see. And when we do that, these bands of DNA will actually move and migrate down through the gel. So here's a scientist, just like what we're doing. Here's our gel. They're squirting and inserting little cut-up samples of DNA into this gel. And when we do that, what we're going to see is that smaller pieces of DNA, smaller chunks, will move farther within the gel than larger ones. And we'll actually end up with, if we were to put DNA into this gel and turn it on here, each of these little bands are fragments or chunks or pieces of DNA. And they'll move down through the gel. Now, the smaller the size is of the fragment will move farther. The larger ones will move a little more slowly. And we end up with what's called a DNA fingerprint. In essence, we end up with this banding pattern. So this sample of DNA, wherever it came from, when cut up by a certain enzyme, will be cut into 4 pieces. Someone else's DNA has different sequences, different bases, will be cut slightly differently and will have a different pattern. Pretty simple. So we'll be doing more with gel electrophoresis in class and some sample activities, but going back again, just as though we did in our video with this cutting and pasting, if you will, just as we saw in the previous one, as long as we have sticky ends that match up, we can create recombinant DNA. We can actually, for example, notice we've cut a human gene here with a sticky end and mouse DNA. A mouse is different from a human, but their DNA is made of the same basis. So as long as we have these sticky ends, they could actually match up together. So we could create an actual piece of DNA that's made up of both human DNA and mouse DNA, which is crazy. We can take different genes from various plants and mix them. We're unlimited, really. And as we can see in this example down here in B, here's a bacterial DNA or a plasmid from E. coli, a type of bacterium it's in our gut. And we can, as long as we cut, for example, a gene on how to make insulin, as we saw earlier in our DNA unit, as long as we cut them the same way so their sticky ends match up, we can actually put a human gene into this bacterial DNA so that now that bacteria can make insulin, which is a trick that many biotech companies, pharmaceutical companies will do to mass produce insulin. Because as these E. coli reproduce and they transcribe and translate their genes, they're going to produce insulin for us. A great trick. We can make these bacteria be our slaves, as we saw. So overall, before we start playing with these things in class again, these tricks, remember, we can analyze human DNA with gel electrophoresis to do crimes, paternity cases, etc., to compare DNA. We can make transgenic species, which, as we said, could make a glow-in-the-dark plant, or bacteria with human genes, or humans with mouse genes, etc. We're really unlimited on what we can do with that. Actually, very, very crazy in that. Some of our tricks, etc. So, as we said, like for example, to end, here's a transgenic plant. This is actually, I believe, a tobacco plant in its earliest stages, but notice it's glowing in the dark. What scientists did here, it's called bioluminescence. They've inserted a gene from a firefly. You know, the Heine-lighters lighting up at night, their abdomens producing an enzyme called luciferase, which can bioluminesce and cause a chemical reaction, flashes of light. That gene was inserted into embryonic cells from this plant. As those cells reproduced and copied themselves, they also copied that gene. So all of the cells in this plant's structure, its organism, its being, have the ability to produce that enzyme luciferase. So that plant can now, in essence, almost glow in the dark, can produce this bioluminescence. Very, very crazy. And just another quick example of one we'll look at. Here's a sample gel electrophoresis from a murder trial. And without going into too many specifics, because we're going to try this in class, here is the defendant's DNA sample cut with a restriction enzyme. Notice the banding pattern here, the pieces of DNA, smaller pieces down lower, larger pieces up here. And here's DNA samples from, excuse me, this is from the defendant, here is from the victim. And here is, let's say we arrested someone that had blood on their shirt and we think they may have killed this person. What we do is we cut up the victim's DNA and run that with all these bands, and then we take blood samples. There's going to be DNA in the blood samples. We cut that and run that, and then we do that for the defendant, the person that, oh, that wasn't me. And if we look, we can see some matches here. This defendant's DNA is not the same as the DNA that was on their shirt. What's happened here is that if we look, the DNA from the victim, the person that was murdered, matches exactly the DNA on this defendant's shirt. In other words, we've proven that the blood, the DNA on that defendant's shirt, was not the defendant's. It came from the victim. Notice there are some similarities here, that they have, people do have some overlap in some of their DNA, but we're looking for exact matches here. And in this case, the blood on the shirt matches exactly the victim. So we'll be trying that in class as well. And then also, that's going to open up, lastly, what we'll look at later on next week. What about ethics? Just because we can do these tricks, should we? Should we clone organisms? Should we create new species, transgenic species, like glow-in-the-dark plants or glow-in-the-dark mice? We can do it, but should we? Should we modify DNA so we create super crops or super animals, like, let's say, cows that produce 30% more muscle, more meat, or corn that can survive drought-like conditions? Sounds awesome, but could there be any problems with that? There are a lot of people that are against that. So just be thinking about any scenarios that come into mind with that, and we'll be looking at that in class later on. So for now, we've introduced it. Now it's time to put these into practice and play with these ideas in class.